All right, so our first function is sensory input, and we've got receptors that detect changes in stimuli. So this can be anything from a change in temperature, to a change in stretch, to a change in chemical concentration. We'll talk about all of the different kinds of receptors at the end of the semester when we talk about senses. We'll talk in general about receptors today, but what we can say for this first function of the central nervous system is that we have receptors that are going to detect changes in stimuli and communicate the information to the central nervous system. So that's our first function, is having these receptors that are detecting changes and communicating it back to the central nervous system. The second function is integration. The central nervous system is doing this. It's taking in all of the information that's coming from every sensory receptor out in your body, it's integrating it and deciding what to do about it, if anything. So for integration, we can say that the CNS takes all incoming info and decides what to do about it. If we've got to do something about it, then the third function will be carried out through what we call motor output. And so the central nervous system will stimulate effector organs that bring about some change. So our third function is motor output. And these are commands from the central nervous system to what are called effector organs. That are going to bring about some change. So effector organs are going to be able to carry out this change, and in the case of bringing information out of the sensory nerve or the central nervous system out to the peripheral nervous system, we've got two types of effector organs. Muscle, that can be smooth, that can be skeletal. If we need to change heart rate, it can be cardiac. The heart is kind of weird though. It sets its own pace, and the central nervous system only ever does anything to it if we need to alter that pace. So all three types of muscle would be effector organs, and then glands can also be stimulated by the nervous system. All right, so for our function, we get some kind of sensory input. In this case, we look at a glass of water. The input is this visual stimuli that's going to be communicated back to the central nervous system that integrates it, takes in all the incoming information, decides what to do about it. You see the water, it makes you thirsty, so your central nervous system will send motor output to effectors to make you go get a glass of water and drink it. Okay, then we divide our central nervous, or our nervous system into two regions structurally, and then we can further divide the peripheral nervous system. So if we look first, our first division is the central nervous system, and this contains our brain and spinal cord. So our first division is the central nervous system, and the organs of the central nervous system are the brain and spinal cord. So these would be our structural divisions. We'll look at functional divisions in just a second. The other structural division is what's called the peripheral nervous system, or the PNS. And this is everything outside your brain and spinal cord that's responsible for bringing in sensor information or carrying out motor output. So the organs of the peripheral nervous system are nerves. And we've got two types, those that exit the brain, that are called cranial nerves. And we have spinal nerves that exit the spinal cord. The other parts that are part of the peripheral nervous system are our receptors. So those sensory receptors that are going to pick up and detect changes in various types of stimuli. So we've got receptors. A lot of books, I think including yours, doesn't include it, like in your picture of the peripheral nervous system, but if receptors aren't part of the nervous system, I don't know what they're part of. So they definitely are part of the peripheral nervous system. They're detecting changes. So again, if we look at our structural divisions, our central nervous system is our brain and spinal cord, which is housed in the dorsal cavity and protected by membranes called meninges. And then our peripheral nervous system are the cranial nerves that exit the brain and the spinal nerves that exit the spinal cord. What this is showing you, you can see this thing called a ganglia here. These are collections of cell bodies outside our spinal cords, but we'll talk more about those when we talk about our neurons. Okay, so then if we look at functional differences, or the way that we can divide our peripheral nervous system based on function, is our peripheral nervous system has an afferent division or a sensory division that's bringing in sensory information, a motor division or an efferent division that's bringing out our motor output, and then we can divide that further. So for our peripheral nervous system, we've got a sensory division. This is called afferent because it's bringing in information. So anytime you see afferent, it's going into something, or I always think approaching, afferent, versus our motor, or efferent is exiting. So these are our two divisions. And in our sensory and our motor divisions, we have two branches. Those branches that we're aware of, so we call that somatic. So in our sensory division, this would be somatic. And this brings in senses that we're aware of, like a change in temperature or a change in stretch. You taste something on your tongue. If you're aware of it, it's part of the somatic sensory division. So we'll say this is senses you're aware of. The other division of our sensory, or the other type of sensory information that we can get, our central nervous system gets it, but it's below our consciousness. This is called visceral sensory information. And so this is things coming like from your viscera, your organs. So level of stretch of your blood vessels would come in through somatic or visceral sensory pathways. Or like the level of the pH in your stomach. That's going to be picked up by chemoreceptors that are below your conscious awareness. So we'll say this is sensory information below your conscious awareness. For our motor, our efferent division, we also have two branches. The somatic nervous system that we're in control of. So our somatic output would be just to our skeletal muscles. So our somatic nervous system, and this is the motor output that you have control over. So it's going to specifically innervate our skeletal muscle fibers. So these skeletal muscles that bring about voluntary movement, or that control our voluntary movement. And the other division of our motor division is what's called the autonomic nervous system. We're going to spend a lot of time at the end of the semester talking about this. It is really important because it controls all of the stuff we're going to talk about next semester. So your autonomic nervous system is abbreviated your ANS, and this controls everything that you're not aware of, or that you don't have control over, like your heart rate, like secreting things in your digestive system, causing motility in the digestive system, changing respiration rate. All of those things are part of the autonomic nervous system. So this controls motor output that you don't have voluntary control over. So the targets in the autonomic nervous system would be your cardiac muscle, your smooth muscle, and your glands. So we'll say it controls output you don't control, or you don't have control over, voluntary control over. So the effectors are smooth muscle and cardiac muscle and glands. There are two branches of the autonomic nervous system that we're going to spend a great deal of time talking about at the end of the semester. And again, that's because this is really important in controlling all of the organ systems you're going to talk about next semester. So this figure in your book, 11.3, kind of breaks out the organization in different ways that we can look at it. So our central nervous system, again, is our brain and spinal cord. It acts as the major control center for most stuff. Then our peripheral nervous system is our cranial nerves, our spinal nerves, and our receptors. And it's going to be communicating information back and forth from those receptors to the central nervous system. We can divide it into the sensory afferent division that's bringing in our sensory information from these receptors. So our somatic is bringing in information that we're aware of. The visceral is bringing in information that is below our conscious awareness. And so it's taking information from those receptors to our central nervous system. If we have to do anything about any of that, we've got our motor division, or the efferent division, that brings down commands from the central nervous system to the peripheral nervous system. Again, our motor division is branched into the somatic nervous system that you have voluntary control over. So the only effectors in the somatic nervous system are your skeletal muscles. It's the only thing you have voluntary control over. Your autonomic nervous system controls everything else. So this is our visceral motor output that's involuntary, and it's going to conduct impulses from our central nervous system to cardiac muscles, smooth muscle, or glands. And again, it's got two divisions that we'll talk about at the end of the semester. I'll just briefly introduce them now because they're on this figure, but it's divided into what's called our sympathetic nervous system and our parasympathetic nervous system, with the SNS and the PNS. Our sympathetic nervous system is in control of like our fight or flight responses. And our PNS, parasympathetic nervous system, is in control of our rest and digest responses. So we'll talk more about that at the end of the semester. Are there any questions over the breakdown of the organization so far? Okay, so nervous tissue. In order to be tissue, we have to have more than one cell type. So if we look at our nervous system, it's got two main categories of cells. First, our neuroglial cells, and our neuroglial cells are supporting cells. So these are supporting cells, and we're going to talk about the specific types in the CNS and PNS in just a minute. The other type of cell that we have in the nervous system is what most people think of when they think of the nervous system. These are neurons, and these are the signaling cells. So they conduct action pulses all around the nervous system and transmit information from one place to another. So they get all of the attention, but our neuroglial cells are very, very important. So in our central nervous system, we have more glial cells than we have in our peripheral nervous system. So if we first look at our CNS neuroglia, our first types of cells are these cells called astrocytes. And these are the most abundant neuroglial cells that you'll find. They get this name astrocytes because they kind of look like stars. And so you can see this is a neuron back here, and the astrocytes are found between our neurons. They support our neurons. Really what they do is they help kind of give structure. They also form the blood-brain barrier. So this is a capillary that you see coming through here. And these astrocytes have these extensions that wrap around the capillary and create the blood-brain barrier. So that's an important function. So what we can say about them is they're the most abundant. They provide structural support. They form the blood-brain barrier. Another thing about them is that, I guess it kind of fits in with this providing structural support, but they fill in the space of dying neurons. So if a neuron is dying off, your brain still needs to maintain its shape, so astrocytes will take over and fill in that space. A couple things just to think about. Well, what's a blood-brain barrier? Why do we have it? The blood-brain barrier protects the brain from the passive passage of materials from your blood into your brain. And so it keeps most stuff out. And our brain and spinal cord and our nerves are all covered in thick connective tissue membranes. In your brain and spinal cord, it's called meninges. We're going to look at the meninges starting next week that have 3 layers and all of this. Your spinal nerves have connective tissue sheaths on the outside. And so your nervous system never actually is in contact with your immune system. So your blood-brain barrier helps keep pathogens and things out. We'll talk about other glial cells that act for immunity in just a minute. But the blood-brain barrier is really going to keep most stuff out of the brain. And what about this other function of astrocytes, that they fill in the space of dying neurons? Why do we have dying neurons? You've probably heard the expression that you only use 10% of your brain. you actually use 100% of your brain. You just use only about 10% of its maximum capacity. And that's because you're born with way more neurons than you can ever use. So nervous tissue is hard to grow, it's very metabolically active. So when you're born, you start off with all the neurons you're ever going to have and more. And what happens is at prescribed intervals, like at 3 years old, at 5 years old, at 8 years old, at around 11 years old, you undergo what are called neuronal clippings. And what happens is you kill off all the neurons that you're not using, and you solidify the connections that you are. So when that happens, our astrocytes fill in the space of those neurons that we're killing off, so that your brain doesn't lose its structural integrity. So the astrocytes are really, really important as far as supporting the neurons in their development and basically helping to just kind of keep it all together. So the only immunity that the central nervous system has is provided through what are called microglial cells. And again, this is because your neurons and, well, your brain and your spinal cord are all ensheathed in meninges. And so really, if there's any exposure of the immune system to the nervous system, it's a bad thing. So, because since they're all sheathed in connective tissue, your immune cells don't recognize neurons as self. So as soon as you get like nerve damage and you like break through that connective tissue sheath, you'll actually start an immune response. So anytime there's any kind of damage to that connective tissue and your immune system sees it, then it'll start an immune response and cause scar tissue in your nerve. So the only immunity that you have in your central nervous system comes from these cells called microglial cells. And what they do is they're phagocytic for anything foreign. So they'll just gobble up anything that they don't recognize as self. So if you get weird protein fragments or something that gets into your cerebrospinal fluid for some reason, then your microglial cells can gobble it up. They can gobble up those dying neurons and get rid of the debris. So the microglial cells provide the only immunity for the central nervous system, and they do it by being phagocytic. What do they eat? They eat up any foreign particles that make it through, and any debris from any dying cells. So our next type of microglial cells are these cells called ependymal cells. And ependymal cells form an epithelial-like lining of some of these spaces that are called ventricles in our brain. We've got some fluid-filled spaces in our brain that are full of this fluid called cerebrospinal fluid, and ependymal cells line that area, and it's called choroid plexus, and this is the only place in the brain that's free of blood-brain barrier. And that's because our ependymal cells have to be able to get the things that they need to make cerebrospinal fluid out of the blood and make it and secrete it into the ventricles. So our next type of cell in the central nervous system are these ependymal cells. And we can say that they form an epithelial-like lining in this region called the choroid plexus. So your brain has these fluid-filled cavities called ventricles. This is a model of the ventricles, and they're full of this fluid called cerebrospinal fluid. And cerebrospinal fluid is important because it helps maintain ion balance in the brain, and it helps to keep the brain buoyant. And all of these little pink areas, they're kind of hard to see, but when you look at your models during lab, make sure you look at them. All of these little pink areas lining our ventricles, I wonder if I can pull this off, are choroid plexus. And these are areas that are free. Oh, shoot. I just fixed this thing, but I just broke it. So now we can see our choroid plexus. So it's this pink area that lines the ventricles, and it has these ependymal cells that are in contact with our blood capillaries, and they're pulling things out of the blood that they need to make cerebrospinal fluid. So they form this epithelial-like lining in this region called the choroid plexus, and their big job is to create cerebrospinal fluid. The last type of cell that we have, of neuroglial cell, is what's called an oligodendrocyte. And our oligodendrocytes are these cells that are myelinating cells in the central nervous system. So this is its nucleus, and what they do is they reach out and they branch around neurons and surround it in this stuff called myelin. So these are myelinating cells in the CNS. Well, what's myelin? It's this special, like, insulating material that helps to speed up conduction of our nerve signals. We'll talk more about it in a little bit. So in our peripheral nervous system, we have just two types of glial cells. The first are called satellite cells, and they wrap around the cell bodies of neurons that are running in nerves, and they kind of act like astrocytes. So they're helping to kind of form this barrier between the cell body of a neuron and a nerve and the interstitial fluid, so that not everything can just passively pass through it. So these are like astrocytes. And then our other cells are called Schwann cells, or neurolemmocytes. And these are the myelinating cells in our peripheral nervous system. So if we look at this figure, this is showing us a neuron that's running in a nerve, a sensory nerve, and it's got this cell body right here that's protected by our satellite cells. So it's cut it away so we can see our cell body, but the satellite cells totally surround the cell body. And they're helping to make sure that not everything can pass passively back and forth between the neuron cell body and the interstitial fluid. And then our Schwann cells, each one of these blue things that you see is a myelin sheath created by a Schwann cell. So what's a myelin sheath? You've probably heard of MS, multiple sclerosis. And what happens in multiple sclerosis is these people can't make complete myelin sheaths. They have thin myelin sheaths or no myelin sheaths. They lose their ability to myelinate. And what happens in that case is your nerve signaling slows so far down that it's really hard to execute motor output. So we'll talk a little bit more about it in a little bit, but basically that's the point of myelin, is that it insulates the nerve and it speeds up the conduction of your action potentials. So if you think about it, your sciatic nerve runs from the bottom of your spinal cord, there in your sacrum, all the way, it goes through your sciatic notch and it goes all the way to the bottom of your foot. It controls everything in the bottom of your foot. It's the biggest nerve in your body. I can wiggle my big toe as quick as I think about it. So as soon as I think about it, my big toe is wiggling. That's a long distance for an action potential to travel. And it can go so quickly because of these myelin sheaths. So we'll talk more about it in a little bit, but just kind of put that in your back pocket for now. That's what myelin is good for. It speeds up the conduction of action potential. The other types of cells in our nervous tissue are neurons. These are the cells that are doing all of our conducting. And what are special characteristics of neurons? We've kind of already talked about one, which is extreme longevity. And that is that you're born with all the neurons you're ever going to have. So when you die, the neurons that you have been with you your whole life. So basically, they live a really long time. So you die with neurons you were born with. So that's a special characteristic. Another characteristic is that they are amitotic, with just like one exception. This amitotic means that they do not undergo mitosis. So you're born with all you have, and then, like I said, you start killing them off as you age. There's one exception. Deep in your brain, where you're forming new memories, in this area called your hippocampus, it brings in all of this new information and then ships it out to where it's got to be held and processed in the brain. When you first are learning a new memory, deep in that hippocampus region, those neurons undergo mitosis. Which kind of makes sense. If you're learning new things, then you need to be making new connections. So that's one exception to this rule of amitotic. The other thing that's special about them is that they are highly, highly metabolic. This is also what makes them such a costly cell to have. They require a continual input of glucose and oxygen, even when you're sleeping. So they have a high metabolic rate. They require continuous glucose and oxygen. So this is so important that you actually have hormones that, like, if you have low blood sugar or you're stressed, you have hormones that will break down your skeletal muscle so that we can use those proteins for gluconeogenesis. So if you think about that, breaking down skeletal muscle is probably not a good idea. But it's so important for your brain to have glucose and oxygen that if you're stressed and you need it, then that's what we'll do. Just, okay, well, skeletal muscles aren't vital, but your brain is. So they will, when glucose, your stress hormone, is on board, it will break down your skeletal muscles so that we can liberate those proteins for gluconeogenesis. So that is really it's a special characteristic, it's unique, and it's very, very important for normal neuron functioning. So what does a neuron look like? It's got different regions that are going to be doing different things. It's got a cell body that's kind of like this enlarged portion of it. So it's got a cell body that's also called a soma. In the central nervous system, where we have groups of cell bodies gathered together, we call them nuclei. So we'll say groups of, I'm going to just write somas because it's quicker. In the central nervous system are called nuclei, and in the peripheral nervous system are called ganglia. So that one picture we looked at showed ganglia outside the spinal cord. You can also see them on this model. So this model has spinal nerves exiting through those intervertebral foramen. These little enlarged regions that are just lateral to the spinal cord are the ganglia that have all the cell bodies of our sensory nerves that are coming in. So in the brain we call them nuclei. In the peripheral nervous system we call them ganglia. So those are groups of our cell bodies. What are other parts of neurons? The transmitting part of a neuron that leads away from the cell body is called an axon. We met our axons a little bit for the skeletal or the muscular system. So this part here that's leading away and conducting our action potential is the axon. And again, when we've got axons running together in the central nervous system, we call them tracts. And then axons running together in the peripheral nervous system, we call nerves. So your axons running together in the central nervous system are called tracts, and in the peripheral nervous system are called nerves. And the last part of a neuron are the receptive segments that are taking in all of the input. They're called dendrites. So they bring information to the soma. So in the nervous system, a dendrite would be communicating with some other neuron. So you'd see an axon terminal synapsing onto the dendrite. So this axon terminal down here is synapsing onto another dendrite. So this is our receptive segment. So if we look at a general schema of a neuron, again here is its soma, or the cell body here with its nucleus. It's got all these dendrites hanging off of it that are receptive regions. The soma can also have axons synapse onto it, so it's also receptive. Then leading away from the soma, we've got the axon, which is conducting away our signals. We've got different parts of the axon that we'll go ahead and label now. This enlarged region right here is called the axon hillock. This is where we're going to sum up all of the input that's coming into our dendrites and cell bodies and decide whether or not to shoot an action potential. So we'll say this sums up input and fires our action potential. So the action potential is conducted down the length of the axon, and then when we get to this bottom region here, this would be our axon terminal. The branches are called telodendria. The enlarged regions at the end have different names depending on what book you look at, but they can be called synaptic boutons. Let's see if I thought I had a picture. No, I don't. Okay. So you can see them called synaptic knobs, axon, I think it's axon bouton, which bouton is the French word for button. They kind of look like buttons. Some books just call it the axon terminal, although this kind of whole end region is the terminal of the axon where it's terminating. Axon hillock. It sums input and fires the action potential. So that's the structure of the axon. It's got the axon hillock, this enlarged region, where we've got our voltage. This is the first place we see voltage-gated sodium channels. So if these other channels up here that we'll talk about in a little bit, neurotransmitter-gated channels, open and we get enough positive charge in here to trip these voltage-gated channels, then the action potential is going to conduct down the length of the axon and you can't stop it. So, but we'll talk more about that in a little bit. So then this is the majority of the axon is spreading away and taking the message down to the axon terminals. These branches again are called telodendria, and the enlarged regions are our synaptic knobs or axon boutons. And this is going to be where we're talking to the next dendrite. So the function of the axon is to conduct the action potential away from the soma. So the last part of the axon to be aware of is the myelin sheath. And you'll see these myelin sheaths that surround the axon. And in between it we have your book, what your book calls the myelin sheath gap, or the axonal gap or something like that. Most books call it, or well, I guess maybe they're trying to move away from it. Most books that I've used call it the nodes of Ranvier. Even I think the videos that come with this book call it the node of Ranvier. This is your, or you could call it the axon gap or the node of Ranvier. It's the space between our myelin sheaths. And again, our myelin sheath is insulating the axon. So if you look in our peripheral nervous system, this is showing us Schwann cells. Our Schwann cell, it's a cell, so it's got a nucleus here, but it'll start wrapping around and around and around and around and around this neuron. And as that happens, the nucleus gets pushed to the outside, and then you've got these several layers of myelin that form the myelin sheath. And again, this is going to be important because it speeds up the conduction of our action potential. So I'm actually going to skip ahead for a minute because I added these pictures from the end of your book, or the end of the chapter is when it describes myelin sheaths or how they're working, but I feel like it doesn't make a lot of sense to talk about them if you don't know what they're doing. So if you think about an action potential, it's this change in where we go from positive charge to negative charge, and if we look at the inside of the cell, it's usually sitting a negative at rest. So if we have this influx of positive charge and we don't insulate it, then our sodium-potassium pumps and our lead channels take over and you lose the integrity of that signal. So this is showing a bare plasma membrane, and if you stimulate it so that we get this influx of sodium right here, the further you get away from the area of stimulus, the more your signal decays. So because those pumps and channels are going to take over, sodium is going to leak back out of the cell, it's going to leak into the cell, we're going to have potassium leaking out, more potassium leak channels, so it'll bring it back negative. So what's happening in these regions like our dendrites, where we don't have these myelin sheaths, then the further you get away from the area of stimulus, the more your voltage decays. So those aren't long lasting and they can't travel long distances. If we look at the axon, so that was showing us a dendrite where we've got no voltage gated channels and we've got no myelin sheath. If we look at an axon where we do have voltage gated channels, but this is an unmyelinated axon, then if this area where we get a stimulus, our voltage gated channel opens and it'll allow that sodium to rush into positive 30, that's okay, and we can conduct the signal as long as our next voltage gated channel is close enough that our signal doesn't decay. So if this is positive 30 here, our pumps and things are taking, and leak channels are taking back over in here, so you're going to lose some of that signal. Doesn't matter as long as you still have that negative 55 threshold to open this next channel. But basically, since the charge is decaying between here, it takes a lot longer for this signal to be conducted. Because now we've got this less of a signal that's like decaying, but I mean, as long as it's still reaching threshold, we can open this next voltage gated channel. I mean, we do have unmyelinated axons, but they're just not very fast. In a myelinated axon, what happens is if we look here at our voltage gated channel, let's say it's at our axon hillock, then we'll have sodium rushes in, and now potassium's not going to be leaking out through these leak channels because we're insulating it. So you're maintaining the strength of that action potential until we hit this Myelin Sheath gap, or the node of Ranvier, where we'll have another voltage gated channel. So now that can open up, and our sodium can dump in, and now we won't lose the integrity of that signal as we transmit down it. This creates what we call saltatory conduction, and it really speeds up the conduction of our nerve impulse. And if you know anything about like copper wire, they insulate copper wire for the same reason. That if you have uninsulated wire, it loses charge to the external environment and it slows down your signal. If you just put some rubber around it, then it insulates it and it can maintain that charge and speed up the conduction of your signal. So that's what myelin is doing for the axon. It's keeping this nice and insulated so that we don't lose our charge through those leak channels. And basically what happens then is you regenerate your action potential at every node. So it hops down the length of your neuron from node to node to node. And it really makes it go a lot faster. So with all of anatomy and physiology, we classify things based on structure and based on function. So first we'll look at the anatomical or structural classification of neurons. Our first type of structural classification is what we call multipolar. And this neuron that I've drawn over here is a multipolar neuron. So poles are different ends. And if you look on this end, we've got a bunch of dendrites. On this end we have one axon. You'll always only have one axon. But it's multipolar because we've got a lot of dendrites on one side. Multipolar neurons are the most abundant neurons that you have. So these are the most abundant. And they have many dendrites and one axon. Now the axon may branch, but there's only one axon leaving away from the cell body. Bipolar neurons are the next type of neurons that we have, and these have two poles. They've got one dendrite coming off and one axon coming off. Now the dendrite might have branches, the axon might have branches, but if you look at the cell body, there are only two things coming off of it. One dendrite and one axon. We find these in places like your eye, or your tongue, or your olfactory epithelium. So these are going to be important when they're hooked up to a sensory receptor. But we'll talk more about that in just a minute. And then our last type are unipolar. And they just have one extension that's mostly axon coming off of it. So we'll just say for now, one extension from the soma. And if we look at these different types of neurons, by structural classification, here's a multipolar neuron. Again, it's got many dendrites coming into the soma, one axon that's leaving. Sure, the axon branches, but it's just one leaving away from the cell body. In our bipolar neurons, we've got a dendrite. It's got branches, but it's just one dendrite coming into the cell body, and then one axon extending away from the cell body. And then in our unipolar neurons, these are what we find like in our sensory neurons that are bringing in sensory information into our central nervous system. They've got one extension that is mostly axon. So it comes off, and we've got this axon that branches out to the periphery. It's got little receptive endings that are in contact with the sensory receptor. So out here could be like a hot receptor. And the hot receptor is right in contact with these receptive endings, and it will communicate this information about that we're hot really quickly along this axon, and then it'll go back into the central nervous system and branch into what are called interneurons to bring that information to the central nervous system. So we find these like in your sensory nerves, and they are really fast conducting because you don't have to sum up anything about the information coming in. You just can shoot past the cell body and bring it right into the central nervous system. So they've got one extension from the soma. It's mostly axon, but it's got some receptive ends. So this is showing just different types of multipolar neurons we can have. You can have really a lot of dendrites coming onto it, or just a couple. Our bipolar neurons, again, we've got one dendrite extending away. It may branch, but it's still just one dendrite that's attached to the cell body, and then one axon that conducts away from it. Unipolar are going to be most of our sensory neurons in our peripheral nervous system that are bringing information to our spinal cord are unipolar. So they've got these receptive endings hooked up to a sensory receptor, and then they bring that information along that axon to the central nervous system. Yeah. We can also classify them based on physiology. So if we're looking at the function and how we classify them, we've got sensory neurons, and these are bringing in sensory information from a receptor. So most of like your senses that you feel in your skin, like heat, pain, pressure, those are coming in through sensory neurons that are unipolar. But sensory information coming into your eye, or coming into your nose, or your tongue, those come in through sensory neurons that are bipolar. When we go through the senses, we'll talk more about the specifics, so be aware that even though they have both classes based on what they look like and what they're doing. So our sensory neurons, by functional class, are just bringing in sensory information from some receptor, but we can have different types by structure. Then our second type based on function are motor, and these are, I don't know if I want to say always multipolar, but they are by far mostly multipolar, and these bring motor commands from the central nervous system out to the peripheral nervous system. And then the third type that we have based on function are also multipolar by structure, and what they're called are interneurons, and they are the link between our sensory neurons and our motor neurons. And they are totally contained in the CNS. So if we think about sensory neurons, if they're bringing information in from the receptors to the central nervous system, then they've got a part that's out in the body and a part that's in the central nervous system. If you think about motor neurons, if they're bringing commands from the central nervous system to the peripheral nervous system, they also have a part that's in the central nervous system and a part that's in the peripheral nervous system. Interneurons do not. They are totally contained in the central nervous system. So this is showing us an example, or when we can find the different types of functional classes. So you can see here, both of these are multipolar neurons. This is an interneuron in the spinal cord that is bringing information from your ascending, like higher brain centers out to a motor neuron. So the interneuron's totally contained in the spinal cord, and then it's sending output to this motor neuron that's going out to a muscle. This would be a sensory neuron. It's bipolar by structure, sensory by function. It's bringing in light information from your eye and conducting it back along your optic nerve into your brain. And then this is a sensory neuron from your skin, and these again are unipolar. You can see its body here, and then it's got the one extension that branches into a big long axon that's in contact with some sensory receptor in the skin, and then it's got this central process that's going to go bring this information to an interneuron in the spinal cord.